Dr. Jane Roskams is our guest. She is a stem cell expert. She works out of the Brain Research Center. She's a professor in the Department of Zoology. Zoology and stem cells. I'm trying to make the link.
There isn't.
Okay.
I shouldn't say
Neuroscience, I would think.
Yes, absolutely. So I'm a neuroscientist, but my appointment is in the Department of Zoology. And I shouldn't say there isn't a link, because every organism on the planet, I shouldn't say every organism, but every vertebrate and many invertebrates have stem cells. And we can actually learn a great deal about regeneration from looking at organisms that can regenerate more successfully than we can. So there is a zoology link. My main link with it is that's the department that I have an appointment in and do my teaching through.
Sure. So in the lab, when you harvest a stem cell and you're doing some research with it, and what do? Take the stem cell from the brain or the spinal cord? How does it work?
Good question. It depends on the question that you're asking. So ideally, if you think you've got a stem cell that's like the ones that exist in the embryo, you want to be able to make every cell in the nervous system with it. But for the most part, when we're talking about disease states or injuries, we don't need to make every cell in the nervous system. We need to make the small number of cells that have been damaged or that aren't working normally. So we don't need to have access to stem cells that will make everything. We need to have access to cells that can make the cells we need. So what we do is we test. Can the cells that we have that we can isolate and put in a culture dish, can we push them and force them to make the cells that you need, whether it's new neurons, the cells that support the rest of the neurons in the nervous system, glial cells, the ones that make coverings and wrappings for tracts to work properly. These are the questions that we're now asking with the new cells that we've identified because of the Allen Project collaboration.
If you have a disease such as multiple sclerosis or ALS, Lou Gehrig's disease, if you have something that has to do with the wrapping of the nerves, I think that has something to do with it.
Absolutely.
How do you use a stem cell to make somebody with MS or ALS better?
Right. So the cells that we just identified and discovered for the first time are actually exactly where you'd want them to be able to contribute to repairing those kind of diseases. They're not in the place where everybody thought stem cells existed in the spinal cord, that if they do exist, here's the spot that everybody's been looking in. When we started seeing images come through the Allen Institute project, all of which are now available online, we saw patterns at the outer layers of the spinal cord of genes that should only be turned on in stem cells. And we're looking at these patterns and going, what are they doing in cells around the edge of the spinal cord? And if those cells have some of the capacity of stem cells, we can reach them from the outside instead of having to pierce inside a spinal cord to begin to try and activate them to repair. So what we're doing right now is we're taking those cells out, and because we have their genetic signature, we can identify them individually as different from the rest of the cells in the white matter of the spinal cord. So the ALS and MS largely affect tracts that come through the white matter. And we can put them in a dish, and we can say, in MS, we need to make more cells that will make myelin. So can we do that with these cells? And if we can figure out the cocktail that they need to do that in a dish, we can then introduce that in a very focal, targeted way to areas of the spinal cord that need myelin.
So best case scenario down the road, your dream, or any scientist's dream who works with stem cell research, would be a type of gene therapy, a treatment that would cure MS, help people with MS, reverse the disease?
You bet. So we don't even need to think of gene therapy, because we already know, because of the genes that they have, what they might need, what clues they may need to begin to make more myelin. So those are the clues we're following right now. What we love about finding cell populations like this in a place like the spinal cord, where we didn't think they existed after embryonic life, is that we may be able to just introduce drugs that we've tested in a dish or in a mouse in the lab, to make those cells do what they need to, your own cells that you already have inside your own spinal cord, Right now, we've shown that they do try and become activated and respond when there's a lesion or when you lose myelin. But something about the lesion environment isn't letting them make myelin successfully. So by looking at reducing the hostile environment that isn't letting them do that, and ramping up the environment that will help them make more myelin, or in the case of Lou Gehrig's disease, ALS, make more of the glial cells that will help the surviving neurons act faster. And
so
both
the body will not
begin to progress, and progressively degenerate. If we can just help maintain and help stimulate the patient's own cells to do that, that's what we're looking to do. We know that in many cases it's not because of a genetic defect, so we don't need to use gene therapy to go in and repair. We just need to get these cells to behave a little more similar to the way they did early in development.
Before. Yeah. In a traumatic incident, a quadriplegic, paraplegic, spinal cord injured dramatically, what can it do, stem cell?
The cells that we've discovered, which are stem cell-like, because we haven't shown that they have all the properties of making all the cells in the body. I know
you're working on it.
We are.
Okay.
What's really interesting is in some cases of lesion, we know that these cells try to become activated. We also know from very recent papers out there in the field that in cases of partial lesions, so when you have a spinal cord lesion, it's either severed all the way through or partially, and most spinal cord lesions are partial initially, but the inflammation and swelling extends the level of injury and makes it seem more complete. In cases where there have been some degrees of functional recovery, particularly stimulated by rehab, and we can now mimic these things in a lab with mice and rats, researchers in the last couple of years have noticed that when you get these large groups of tracts that are regenerating, they're doing them across bridges. And the bridges that they've identified have been a bit of a mystery until we discovered the cells that we've discovered. Because they kept saying, they look like the cells that form these bridges and train tracks in the embryo. But these cells don't exist outside the embryo, because nobody had ever shown that they do.
And the politics of it all, as you know, there have been some discussions about whether or not stem cell research in certain cases should even be done. Are we pretty much done with that worldwide, or is it still controversial?
We are reasonably well done with that. I think in all cases, we have to be careful with stem cell research. Where we need to be more careful is in countries that don't have the kind of careful regulation that we have, that they have here. In the US, it's still a little extreme, but during the Obama administration, it's become an awful lot easier to do good, well-controlled stem cell research.
Because the controversy was around
embryos.
An aborted embryo that is not going to live anyway.
Right, absolutely. And most of the controversy was over embryonic stem cells, and what the politicians who were against the idea of embryonic stem cells did was they manipulated the media and they manipulated the public and would show aborted fetuses without actually stating that embryonic stem cells as we know them and make them in a culture dish in the lab actually come from fertilized eggs in IVF clinics.
Okay, so nothing
that bear no resemblance to a human being.
Right. Well, as you know, the press isn't always on it. You've noticed.
Well, and also very clever people manipulate.
And the politics and to do the heavy research as you do. It's great work. It really is. And I read that it's the future of medicine. Cancer treatment on down? Or what?
Absolutely. I mean, the really interesting thing, I think, and that also came out of us looking at the signature of these cells. What's special about these cells? It goes back to your first question. How do you tell the difference? When we look at what was special about these cells and we compared them with other kinds of stem cells in other parts of the brain, we identified a whole brand new small set of genes they use that hadn't previously been linked to stem cell function. And out of 7 of them, 6 of them are genes linked to human diseases.
Really?
Some of which are involved in cancer.
How interesting. Will you come back and talk about that
Absolutely.
one day? after your bike ride and raising those 3 children and all of that, other things that you're doing. So neuroscientist, I can call you.
You bet.
In the Department of Zoology at UBC. Okay, thank you.
You're welcome. Thank you.
Thank you. Nice to see you. Dr. Jane Roskams.